Our last chapter on evolution is dealing with evolution at a larger scale, and this will be our last chapter for our course this semester. I love this joke because I think it summarizes the concepts in chapter 16. This goldfish who just yells evolution and jumps out of the fishbowl. And the reason why I like this cartoon is I think it really encapsulates the misconception or how the concept of evolution gets misapplied or misused. So when we look at evolution, individuals can never, ever evolve. Evolution can only take place at a population scale, at the whole species scale. You need genes to change. You need to see that change played out over multiple generations. So that necessarily implies that an individual can't evolve. So it's a great little cartoon to help us ease into the large-scale evolution patterns that we see. So in order to understand large-scale evolution, we always have to remember that populations evolve, that large-scale patterns affecting change, we have things like climate change, continental drift, those external forces that are working on a different time scale can have a disproportionate effect or can lead to some of the changes that we see in a line of descent. And then we also need to focus on some of those mechanisms that are going to help to keep species separate once we have speciation events occur. So we need to revisit the concept of biological species. Remember that when we're talking about biological species, we have members of a species interbreeding and producing viable offspring. By viable offspring, we mean that first of all, they survive. And not only do they survive, but they can produce offspring of their own. When we're talking about species, we're talking about individuals together that have a shared gene pool, all of those shared genes. Each species is reproductively isolated from every other species. When we talk about species, we're talking about populations. Now, occasionally we will have a whole species be an entire population, but that's for very rare organisms or when we have a species that's probably on the brink of extinction. And of course, when we talk about species, we are talking about individuals with those shared genetics and the idea that species have common ancestry. We can trace that lineage through DNA, RNA, protein analysis, and then also through some of those morphological characteristics. Speciation is when we have the splitting of one species into two or more species. We're going to have gene pool changes. Sometimes it's going to be just a single gene that changes. Sometimes it's going to be the accumulation of lots of gene changes that drive speciation to occur. Sometimes we have really significant differences when speciation has happened. Remember that our robin and bluebird are closely related to one another. They look dramatically different. Our flycatchers, on the other hand, are very similar to one another. So this is showing us that sometimes after speciation, we can have those really subtle differences. Regardless of whether the differences in the species is dramatic or subtle, after the change, those organisms are going to be are unable to interbreed and produce viable offspring. Sorry about this. There we go. So let's look at some of those mechanisms that are going to help to keep species separate. One mechanism is habitat isolation. And what this basically says is that we have two separate species that are preferring or selectively choosing different habitats from one another. That's going to keep them from ever interbreeding with one another. And again, we have flycatchers as a perfect example. The Acadian flycatcher and the alder flycatcher prefer very different breeding habitats. They spend most of their time in Central and South America in the winter, and they come up here to North America to breed. And when they come up here, they're going to selectively choose different habitats to breed. Now you might be thinking to yourself, but wait a minute, I'm seeing some blue and pink overlap there in Ohio and Pennsylvania. Like, how does that play out at that local scale? And I don't know if you've been to those locations, but the eastern part of Ohio and definitely Pennsylvania, where we have the Appalachian Mountains, those mountainous regions we see that same habitat isolation. And it's really strange to be on one side of the mountain and see one type of bird, and then go on the other side of the mountain and see a different type of bird. So even though it looks from our large-scale view here like, well, there should be some overlap in Ohio and Pennsylvania where they're both in the same location. When we get down to that fine grain hiking around in the woods, we see that they're still preferentially selecting different habitats to breed in. Another example of habitat isolation is more modern. It has to do with humans and how we interact in the landscape. So one of the biggest things that we do is we fragment the landscape, and we do this with our roads and our highway expansions. When we do that, we are isolating and separating populations. We could potentially be contributing to speciation events, or we could be contributing to a whole host of things. And sometimes what will happen with the Department of Transportation is they'll get mandates that say, if you're going to expand this project, if you're going to build this highway, you have to make sure that the individuals from the population can still move back and forth. We can still have that gene flow between the populations. You're not isolating them and separating them from one another. So a good example of this is in Madison, Wisconsin. When they were expanding the Beltline, they were going to be separating two populations of Blanding's turtle that are endangered. What they had to do is they had to put big tunnels underneath the highway so that the turtles could move back and forth, and we wouldn't have that habitat isolation occur. Temporal isolation is exactly what you think it is. It's the time when you want to make your babies. So our American toad and our Fowler's toad have different times a year when they want to breed. And that timing, that internal biology saying, now is when I declare territory, now is when I go and find a mate and make my babies, that is what's going to keep these two species separate, even if they live in the same geographic location. Behavioral isolation is what you do to attract a mate, generally, and behavioral isolation can be really fun. A lot of species will have specific things that they do to attract a mate, and that's also going to keep them isolated from all of the other similar species that might even be breeding at the same time. So our fireflies tend to all breed at roughly the same time. And the big question is, if you're a female who's climbed to the top of the vegetation, the top of the grass or the top of the plants in this location, how are you going to know which male out there is of your species, which male you should be breeding with? And what the females are doing is they are looking for flash patterns. If a male flashes a pattern that they recognize as being part of their species, then they will accept that male, mate with him, and the rest is, well, the rest will be evolution over time, natural selection. So behavioral isolation are the behaviors that organisms are doing specific to their species. Mechanical isolation is rare, but very interesting. With mechanical isolation, you literally can't get the bits together. You can't get the reproductive structures together to exchange gametes. And this happens with snails. Snails can build their shell in a clockwise direction, or they can build it in a counterclockwise direction. Depending on which way you built your shell, this is what's going to keep you isolated from other snails when you try to mate. Only clockwise shells can mate with clockwise shells, and these poor snails in this picture and this picture are both having that dilemma where they really want to mate. The hormones are in the water, in the location, in the air. They want to exchange gametes, but there is no way they're mechanically going to get their reproductive structures together. So mechanical isolation can also help keep species separate. Gamete isolation is interesting because it's all happening at that cellular level. And we see gamete isolation happen when the species have a strategy of release all of the gametes, all of the sperm, or all of the egg into the environment at the same time. In other words, the likelihood of species gametes getting together and trying to cross fertilize is a very real consideration. There are a lot of species in the ocean where they just release all their sperm and all their egg, all at the same time. Everybody. You do not want to be swimming in the ocean on that day, but that's their strategy. And if all of the species have all of their gametes floating around, how do you make sure that the sperm and the egg of the correct species get together so fertilization can actually happen? You can actually make the next generation. And so what we find is at the cellular level, generally on the female structure or generally on the egg, there's going to be chemicals that suppress all of the sperm, all of the male gametes of every species except for the species that is the same. So here we have pictures of sea urchins, two separate species of sea urchins that will be releasing all their gametes at the same time. And then my favorite one, seeing how much I love plants, is the way how plants control fertilization when they're getting pollen, which is essentially plant sperm, from all different kinds of plants. So in this picture, the gray structure is the female part of the plant. And we can see multiple species of pollen. We have kind of a tan, spiky ball pollen, that's ragweed. We've got a bunch of kind of yellow and greenish yellow pollen. And then finally we've got this almost looks like a dumpling pollen. And what's happening with the gray female structure is it's releasing a bunch of chemicals to suppress and stop all of the pollen from germinating, except for the pollen that is of the same species. And when that pollen germinates, it makes something called a long pollen tube, and that's where the sperm will swim through in order to in order to fertilize the egg. So gamete isolation is very much happening at the cellular level in these one-on-one interactions between sperm and egg. So all of these are prezygotic isolating mechanisms. What happens if the sperm and the egg of separate species do combine? Do we have any postzygotic isolating mechanisms? And we do. One of those postzygotic isolating mechanisms is very dramatic, and this is zygote mortality. So yeah, the sperm and the egg combined. We might have had a couple of rounds of cell division, but the signaling from the female, the development is all wrong. The gene signaling inside the cells is wrong, and so that combined package of cells is simply going to perish. It's not going to come to fruition. Another type of postzygotic isolating mechanism is hybrid sterility, and we see this with donkeys and horses. Donkeys and horses have roughly the same amount of DNA. They have very similar genes, and so it is possible for a horse and a donkey to mate. When they mate together, they make a mule. The mule's going to have a happy little muley life, but the problem is that mules can't mate with other mules. If you want to keep making mules, you have to keep mating horses and donkeys together. For the most part, almost all mules are going to be sterile. So this is why a mule never gets to be its own separate species. You have to keep making them over and over again by mating donkeys and horses. We have a similar thing with lions and tigers. Let's just pause for a minute here and think about the idea that lions and tigers are never going to come together on their own in the wild. And so if we make a liger, a lion and tiger combination, this is really going to be forced because of artificial situations. Lions are hugely aggressive, and most of the time they're very possessive, mating within their own groups, much less expanding out and mating with a separate species. So if we do happen to mate a lion and a tiger together, you make a liger, they're probably going to have a lot of emotional problems. They seem to be okay physically, but have a lot of emotional situations that make them not ideal pets. Not that a lion or a tiger makes a good pet either, but that's a whole other issue. And if you're going to be making ligers, you have to keep making them over and over again. Most ligers are going to be sterile, which is why we don't have a separate species of liger. Another postzygotic isolating mechanism is the F2 fitness. Now, F2 refers to the second filial generation. So our example here is we can mate a horse and a zebra together, and there's enough genetic similarity between horses and zebras that we can get lots of zorses, and those zorses can have happy little zorse lives. The difficulty becomes when we try to mate two zorses together. The two zorses will make offspring, so you can have zorses make more zorses, but the problem is that the F2, those zorse kids, are all going to be very, very sick and very ill and not make it to reproductive maturity. So in this case then, your horse and zebra can make an F1 generation, the Zorse. The Zorses can make F2s, but those F2s are probably going to die before they can make any more Zorses. And part of the problem here comes down to the zebra. Zebras can have a variable amount of DNA. A horse has 64 individual pieces of DNA. The zebras can have anywhere from like 40 pieces of DNA up to 68. So depending on which zebra your horse mates with, and then how that plays out in the offspring, and you can imagine why by the time you get to the grandkids of the original zebra-horse mating, you're just going to have some problems. So this covers our overview of prezygotic and postzygotic isolating mechanisms, helping to keep our species separate. I don't want to downplay the role of those large-scale isolating mechanisms. So one of the large-scale isolating mechanisms that we talk about is the role of geology and continental drift. Now, the thing we always have to keep in mind is that the plates in the crust are moving very, very slowly. So what this means is that we get a level of consistency, generally a level of consistency in the climate, the precipitation, the temperature, a predictability in seasonal fluctuation. All of the plants adapt to this, all of the animals adapt to this. We get consistency for a period of time. But because we have that constant movement, this large-scale geological transitioning, what that means is that at some point we're going to cross a threshold. At some point our continents are going to get too far apart from one another and animals can't flow back and forth. We get isolation of populations. We get changes in temperature and precipitation. Things don't stay the same forever. But we do get these large-scale patterns of consistency. What we see in the species is that for the most part, species stay relatively the same from generation to generation to generation. We don't get big dramatic changes until all of a sudden something changes in the environment. Then we get these big, huge dramatic changes. So I guess the big message here is things stay the same until something happens, and then all of a sudden we can see dramatic flips, dramatic switches. So when we look at the different types of speciation that we're covering for this class, one of the things to keep in mind is the role of geology and geographic barriers. Our first type of speciation is allopatric speciation. With allopatric speciation, we've got a geographic barrier that's separating two populations. Because these populations are separated, they start experiencing different things. Those external pressures in the environment start to select for different genes, and so over time, over multiple generations, we see dramatic differences. So our example here is we started with a population of pine trees. Because that river was cutting a canyon and separating those two populations, and thus those two populations started experiencing different things, we see the dramatic difference. In this case, one population of pine trees becomes a new species with a lighter coloration of needles. An example in the textbook is of salamanders. In order to understand this example, you first have to think like a salamander. Salamanders are small. They're dependent on water. They are not going to move very far. They like to stay really close to their home. We have to keep in mind how big California is. So when we start with an ancestral population of salamanders in Northern California, each salamander in their lifespan is not going to get very far. So gradually, over time, this population starts to encounter the mountainous regions. The population splits. One population gradually, each generation, multiple generation, hundreds of generations, is gradually walking and moving down south in California, more internally. The other population is closer to the shore. As each generation is going a little bit further, they're experiencing new things, new predators, new disease agents in the environment, different changes in temperature and precipitation and winters and all of those things that start to allow natural selection to take place. Those that have genes that can survive pass them on to the next generation. Those that don't pass on those genes. And so by the time we're looking at populations today, after allopatric speciation has been occurring for a long period of time, we see dramatic differences in the populations of salamanders. We see dramatic differences in the species. Similar thing has happened in the Grand Canyon. The Grand Canyon took a very long time to make. At some point, the population of squirrels, shown at the top of the picture here, became isolated from one another. And what we find is that squirrels on one side of the canyon have that white stripe in their belly, and they've got the white eye ring. While squirrels on the other side are almost completely dark and black. Different environmental pressures, different selective pressures for individuals in those two separate species. Now on the bottom of this picture we have two separate species of ground squirrels. Now to my eye, and probably to your eye, I can't tell the difference between those species. They're so incredibly similar. But when we put them into a container and try and get them to mate, they try to kill each other instead. Chipmunks are pretty aggressive. They are definitely separate species. And it's because there has been enough time where the populations were separated and they were experiencing different things on different sides of the canyon that speciation could occur. Our next example of speciation is sympatric speciation. And here we have some kind of change within individuals living in the same geographic location. So that implies that there's some type of internal signaling that would drive that speciation event to occur, even though they're living in the same location and supposedly experiencing the same environmental pressures. So our first example here is of pine trees, where we see gene changes in some of those individuals led to speciation, which made them have more needles, look fluffier, and have a slightly lighter color green. Another example is of the snapping shrimp. They have experienced sympatric speciation as well, and we're seeing some pretty dramatic phenotype differences. And then these cichlid fish who live in the same lake in Nicaragua. And we can see the two pictures of the fish down on the bottom, that they develop some very different phenotypes. They're looking very different from one another in response to gene changes, in response to this sympatric speciation, because they're living in the same environment. So I like this picture of the main speciation types. The green is our original population, and the yellow shows the individuals in the new population with the genetic changes. Essentially what we're seeing is that transition to a new species. So we start at the top with our green original population, and we can see the role of a geographic barrier and internal pressures starting to lead to that speciation event, where we get two separate distinct species. Now you can ignore peripatric and parapatric, the two center types of speciation. That's something that's covered in higher level ecology classes. And of course, at this point, we have found and named 8.7 million species. We know that there are so many more. We've just barely scratched the surface, especially on things like protists, bacteria, and archaea, and even a lot of the fungi. So of the found and named species, the 8.7 million species, we have that universal tree of life, showing how all the species are interconnected. Yes, they are distinctly different from one another. They all have adaptations that came about for a whole host of different reasons. But this diversity of life is what makes our planet so wonderful and so spectacular. It's part of what makes life on this planet enjoyable, is having all of these very, very diverse species, separate and yet involved in a myriad of different relationships. And this brings us to the end of chapter 16 and the end of our biology class. I will have an end of the semester message that I hope that you enjoy. As always, if you have questions, please email me or stop by Zoom office hours.